What time? Go to his office.
It's an online platform.
Put his number on Canada. Email him. Like, listen, this is ridiculous. Sometimes I have to do things like approve it, you know?
How is it done already though? That's what I don't get.
Yeah. Yeah. But I don't need another like half hour.
Can't think done at one time.
And everybody doesn't have the same pace, you know what I mean? That's part of it.
Thank you.
That's weird.
That's ridiculous that it's only backwards already. Was it fully charged when I got there?
Yeah.
Matt and no comment?
Yeah. Should be.
Yeah, Joe and Kelly are taking it.
But if the time is what's killing you, then you're probably not the only one either. You know what I mean?
And I know it. Mostly. I knew that answer right, because it was just no companies going up. It really
I think that was the class I took online too. Taking one online class. I don't remember that happening. But I definitely email them. And if other students are emailing me too, then you might, yeah, be more likely to listen.
So I think being the honest I was the type of person that like I'm not paying attention. And the second time I'm like,
Did you say
Yeah.
that Monday's
Every time I get
lecture
it.
is going to be on the exam or not?
You say it depends on what we get done today, right?
Yes, that's what I said. And that's why I'm thinking, I'll let you know after, in about an hour. Most likely, yes. Okay. If that's the case, and that's gonna be mitosis, mitotic divisions is the next topic. I like to go through the biological hierarchy, unlike other instructors who do the mitosis and meiosis right before the genetics portion. I do mitosis and meiosis right before after I talk about the cell and before I talk about tissues, animal and plant tissues. So anyways, I'll let you know about an hour. Okay, the cytoskeleton, I mentioned this in the beginning. Microtubules, I think we're all set for the chloroplast. Right, 2 steps involved in photosynthesis, the light and dark reaction. For now, you're going to be responsible for the thylakoid membranes for the light reactions and or the grana. Right, so if I ask a question on an exam, light and reaction of photosynthesis occurs in the stroma, and another choice would be the grana. And you don't see thylakoid membrane, you would assume it would be the grana. And grana is, again, stacks of the thylakoid membranes. So, the dark reaction occurs in the stroma, and that's the gelatinous stuff. They call it the aqueous, mostly water. It's more of a gelatinous goo type of thing. Okay, and the mitochondria is different in both the chloroplast and the mitochondria. Microtubercular lattice, I started this lecture talking about this. Microfilaments, and again, think of them as little thread-like structures. They're fibrous proteins, so whenever you think about fibrous proteins, what should you be thinking of? What type of structure of proteins? Primary or secondary? Okay? The globular proteins would be the tertiary and or quaternary. Okay, so the microfilaments are fibrous proteins. Okay? So again, primary or secondary, usually secondary structure of protein. Actin and myosin, you're gonna see these fibrous proteins and skeletal muscle and cardiac muscle, okay? And any mammal, okay? Actin and myosin. Again, whenever you see IN at the end of a word, that should let you know what type of bioorganic molecule. IN. Right, protein. I think I mentioned that. OSE at the end of a word, that would let you know it's some type of carbohydrate or sugar. Okay, good. Okay. IN, ASE at the end of the word. Enzyme, right. And enzymes are always going to be proteins. But every IN at the end of a word, that refers to protein.
That makes sense. Code says that.
But, yeah, I thought I mentioned it. Myo, you should be thinking muscle, right, good. Unlike the microtubule, right, those would be like globular proteins. And again, you saw in that video the dismantling and reassembling of these microtubules. and that motor protein was holding the vesicle, apparently walking on a microtubule. Tubulin is the primary structure of protein. Found in cilia, flagella, and centrioles. Cytoskeleton, and I showed you black and white image. Again, these are the what you saw in that video. A lot of these fibrous subunits are coming together. And the tubules, the amino acids coming together. Okay, I may have one of the short answers, 5-point short answers. Describe centrioles versus cilia and or flagella. This 9+2 arrangement. Nine plus 0 microtubule triplets. Centrioles. 9 sets. And nothing on the inside. Okay.
Bless you.
These I didn't bring plant cell, animal cell. Where would you see centrioles?
In animal cells?
Only in animal cells, no plant cells. Okay. So every single animal cell, most every single animal cell, not right. I can think of a few. Most of the every single animal cell will have one pair of centrioles. Okay. Of the surround 9 referring to the triplets of microtubules, 9 sets of these triplets, plus 0 on the inside. Cilia and flagella, 9 plus 2 microtubule doublets. Okay. Oh, and these would be connected by these arms. And then there's going to be always two in the center. Okay? And they're connected. The doublets are connected. Only in these microtubule, the flagella and cilia. Again, 9 sets of doublets plus 2 in the center.
2 sets?
9 sets of doublets plus 2 in the center. So that's where you're getting the 9 plus 2 arrangement of doublets in every single cilia and flagella. The reason I'm making a big thing about this is a primitive muscle, the most primitive muscle that we see in kingdom Animalia. Okay? So when ATP is needed, it moves one set of doublets, and these are connected to another set of doublets, and that's why these arms. So this will move one set of doublets, will force all the other sets of doublets to move accordingly. So again, saving energy, okay? Saving adenosine triphosphate, or ATP. The most primitive aspect of a muscle that we see in a lot of the protozoans, okay, in kingdom Protista.
You said you would put that as one of our tests. Can we draw?
Oh
Can we
yeah,
describe it
yeah,
to, like, writing
yeah,
and draw?
yeah, and describe it and draw it. And, right, yeah. 9 plus 2 doublets in flagella and cilia, and, as opposed to centrioles. Okay. Centrioles you'll see again only in animal cells. Actually, that's another main difference between an animal cell and a plant cell. No plant cell will ever have cilia and flagella. No plant cell will ever have flagella and or cilia. You will see little hairs on a plant. Those hairs are not cilia or flagella. They are fibrous forms of a protein.
One more question. The ATP in the flagella one,
Yeah.
what happens in the centriole? It doesn't move that
No.
way?
It moves accordingly with the cytoplasmic streaming and the dismantling of the microtubercular lattice that we'll see when we talk about mitosis,
So centriole
mitotic division.
passive is what you're saying, and cilia flagella active, right? Because one's going with the flow and one is not.
For now, yes, but right, yeah, I don't think of the centrioles as passive energy. They don't need energy. They more of a structural aspect. and I'll tell you after the main theory of why we the plant cells have a very, very rigid cell wall, every single plant cell. So when that plant cell is getting ready to divide, there's protein fibers to grab hold of the chromosomes from one side to the other side. But in animal cells, its plasma membrane is so flaccid that you need some type of structural support. And that is the idea of the centrioles, or more specifically, those centrosomes, where the centrioles will be residing. But again, don't worry about that. We'll get to that later on. But for now, yes, you need ATP or many ATP to get this flagella in motion. But once it's moving, it takes little to no energy.
So what would
To
be
move.
an example of a microtubule in a plant cell because it's only listed as animal cells?
Within the plant cell, you are going to see microtubules and microfilaments, but not arranged in this 9+2 doublets and or not arranged in 9+0 triplets.
Okay, so there's
But
not-
yes, you will see microtubules and microfilaments individually in a plant cell, but not in this arrangement. Any other questions? Okay. Right, yeah, so that's, what I want. Yeah, I'll leave it with that. For now. 9+0, triplets, 9+0 sets of triplets, in a centriole. The 0 again, is referring to nothing on the inside, and sets of triplets. In centrioles, flagella, this is the basal body. The basal body is, this is the plasma membrane. The basal body is inside the cell. I like to think of the basal body as the root of these flagella and or cilia. Again, cilia has the same arrangement of microtubule, 9+2 microtubule doublets as we see in flagella. Again, all of these doublets are grabbing hold, having all these arms on one side to grab hold of another doublet pair, and then another doublet pair. So you need some type of energy to put this in motion. But once it's in motion, biologists always thought that flagella and cilia was beating at the currents within the aqueous solution. But with the electron microscope, we're able to see exactly what, oh, wow, it is not moving with the current. But again, you can see these arms grabbing hold of. But, this would be inside the cell. this, if you think this would be the plasma membrane, this would be the root of this beating flagella.
Can centrioles undulate and stuff?
I'm sorry?
Can the centrioles undulate and
No,
stuff?
no,
Just the-
no. They move in accordance, all together. They're, right, yeah. They can't undulate or move at all, other than as one unit. Oh, and okay, there's always an exception to every, I said no flagellas are ever found in plant cells. I thought about something. The fern, the gametes, half of the male gametes will produce a cell that will have flagella. But that's only one example.
Must have eaten something.
Right, there's always for the most part, right, you're never gonna see, except for that. Fern gametes will have flagella. Okay, cilia, think of paramecium. This would be the nucleus, the Golgi, endoplasmic reticulum, mitochondria, the Golgi body, the paramecium. Paramecium is a genus, genera name. They will have beaten cilia surround, and that's how they move. The male gamete, the spermatozoan, the euglena would be a good example of again, a nucleus, endoplasmic reticulum, mitochondria, the euglena. Well, this would be again the, this would be a flagella. Flagella. Again, all flagellums have this 9+2 arrangement. My point here is that flagella is much, much larger, okay, and more of a whip-like structure than short little cilia. We'll see in our throat cells, cilia, short little hair-like structures. But again, cilia and flagella will always have this 9+2 arrangement. Okay, this is what I was, cytoplasmic streaming, we saw that yesterday in that Elodea leaf under the microscope. All those individual chloroplasts, okay, were moving in accordance with the heat of the lamp of the microscope. Okay? So again, that gave enough energy to drive the gradients, the heat of that. Cytoplasmic streaming, every single cell. Chromosomal movement, we'll see the chromosomes move anaphase, you see a movement. During metaphase, the spindle fibers will attach from either side and literally pull apart duplicated chromosomes. Okay? So again, that would be chromosomal movement. Flagella and cilia I mentioned. Pseudopod. An amoeba would move by a pseudopod. A snail or any mollusk, clam or snails will have a pseudopod. A pseudopod, what is a pseudo? What does pseudo mean? False. What does pod refer to? Foot. So a false foot. Think of a snail moving on its mantle. Okay? But that's mantle is known as a pseudopod. Okay? Amoebas will use pseudopods to move. Cellular movement. Extracellular movement. Okay? Again, you saw that. I pointed that out in that video. Outside of the cell, all those fibers holding every single cell together. Okay? The extracellular matrix. We're going to see the extracellular matrix in the histology portion of this class. Plant and animal cells, tissues. Histology is the study of tissues. The extracellular matrix. So you should be thinking non-living cells. Okay? These are all non-living. Okay? The extracellular, outside of the cell, holding, like the collagen, holding cells together, or the basement membrane holding cells together. That's all non-living tissue. Just the cells themselves. Again, our definition is metabolism. So individual pieces of collagen, fibrous proteins, glycoproteins, and polysaccharides are not living themselves. Together in a cell, that's metabolism. They are able to metabolize.